Hello friends, good evening. Welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about the process of DNA replication in prokaryotes. For example, in E. coli. DNA replication is a biological process in which several enzymes work together to produce an identical copy of a double-stranded DNA molecule. Like other organisms, in E. coli also, the process of DNA replication is divided into three general phases. These phases are initiation, elongation, and termination. First of all, we shall discuss about initiation. The chromosome of E. coli is a single circular double-stranded DNA molecule of 4.6 million base pair. In E. coli and in most of the bacteria, DNA replication starts from a single point of origin of replication, which is known as ORIC. This sequence is known as ORIC, and DNA replication starts from this sequence. ORIC is a 245 base pair sequence. It contains two types of conserved sequences. So, two types of conserved sequences are present in the ORIC. One is 3 repeats of 13 base pair and 4 repeats of 9 base pair. These repeats are known as 13 mers and 9 mers respectively. So, how the process of DNA replication is initiated in E. coli? About 20 molecules of DNA A proteins along with ATP bind with the 9 mer repeats. These are the 9 mer repeats, and the DNA A protein along with ATP bind with these 9 mer repeats. The binding of DNA A protein causes the DNA to wrap around these DNA proteins. In this diagram you see these are the DNA A proteins, and around this DNA A protein the DNA is wrapped. The binding of DNA A protein and ATP trigger the unwinding of the 13 more repeats, thereby causing the formation of an open complex. This is the open complex, and you see here the DNA A protein and ATP have triggered the unwinding of the 13 more repeats. So this 13 more repeats has been opened. After this, two DNA B proteins bind with the 13 more repeats. This binding is facilitated by another protein known as DNA-C. DNA-B and DNA-C interaction causes the opening of the ring of DNA-B protein. The interaction between DNA-C and DNA-B causes the opening of the ring of DNA-B protein, by which this DNA-B protein bind with the 13 more repeats. This is the sequence of 13 more repeats, and here you see these are the two molecules of DNA-B. After binding of DNA-B with the 13 more repeats, ATP hydrolysis occurs and the DNA-C protein is released. DNA-B protein is actually a helicase. For the process of DNA replication, two helicases are required. The function of the helicase is to unwind the DNA double helix. Helicase moves along the DNA molecule in 5' to 3' direction, thereby unwinding the DNA double helix, by which DNA replication occurs. The activity of helicase creates topological stress. This stress causes the unwinded DNA strands to become supercoiled. This topological stress that is induced due to the activity of DNA helicase is relieved by another enzyme known as DNA topoisomerase or DNA gyrase. This enzyme relieves the topological stress by negative supercoiling. After this, specific proteins known as single-stranded binding protein attach to the separated DNA strands and stabilize them. Friends, the enzyme DNA polymerase cannot initiate the process of DNA replication. Thereby, another enzyme known as primase. The enzyme primase catalyzes the synthesis of short RNA sequences known as RNA primer. The RNA primer usually contains 10 to 11 nucleotides, and this primer provides a free 3-hydroxyl group to which DNA polymerase add successive nucleoside triphosphate one by one. So friends, in this diagram, these are the DNA proteins, and around these DNA proteins, this is the DNA that is wrapped around. And this region, which contain 13 more repeats, are already separated. And these are the two helicases. And this complex is known as pre-priming complex. After the formation of pre-priming complex, next step is elongation. After initiation, next phase is elongation. Elongation is the second phase of DNA replication. In elongation, the enzyme DNA polymerase 3 add nucleotides to the free 3-hydroxyl group of a RNA primer. So by adding nucleotides to the free 3-hydroxyl group of a primer, the chain is extended, and this process is known as elongation. In E. coli, DNA polymerase 3 is the primary enzyme that is involved in the process of DNA replication. The sequence of nucleotides that are added by the enzyme DNA polymerase 3 is decided by the sequence of the nitrogenous base in the template strand. So friends, already you know about some of the enzymes that are involved in the process of DNA replication. One enzyme is helicase. The function of the enzyme helicase to unwind the DNA double helix so that various enzymes can get access to the DNA strands by which DNA replication can occur. Another enzyme is primase. As DNA polymerase 3 cannot initiate the synthesis of DNA, so first of all, another enzyme known as primase catalyzes the synthesis of a short RNA sequence known as RNA primer. So the job of the enzyme RNA primase is to synthesize a short RNA sequence which is known as RNA primer. The RNA primer provides a free 3-hydroxyl group to which nucleotides are added by the enzyme DNA polymerase 3. Another enzyme is DNA polymerase 3. DNA polymerase 3 is the basic enzyme. A dimer of DNA polymerase 3 catalyzes the synthesis of DNA during the process of DNA replication. Another protein is single-stranded binding protein. Single-stranded binding protein are the specific protein that bind to the separated DNA strands, stabilize them, and prevent their reannealing. Friends, this diagram explains the process of elongation, and these are the two template strands. These two red colored strands are the template strands. DNA replication proceeds in 5' to 3' direction, and this Y-shaped region is known as replication fork. Replication fork also move in 5' to 3' direction. This is a template strand and this is another template strand. So during the process of DNA replication, Both of these template strands has to be copied by using the enzyme DNA polymerase 3. Friends, in a DNA double helix, the two strands are antiparallel. They run in opposite direction. This template strand run in 3' to 5' direction, whereas this complementary strand run in 5' to 3' direction. And you know that DNA replication occur in 5' to 3' direction. So how a single dimer of DNA polymerase 3 can coordinate the synthesis of these two new complementary strands? Let us see. Friends, during the synthesis of this new complementary strand, direction is not a problem, because this new strand is synthesized continuously in a hassle-free manner in 5' to 3' direction. So the synthesis of this new strand is a straightforward process. This is the RNA primer. This green patch is known as RNA primer, and this RNA primer is laid down by the enzyme RNA primase. After the synthesis of RNA primer, it is the job of the enzyme DNA polymerase 3 to add nucleotides to the free 3-hydroxyl group of the RNA primer, thereby extending the chain. So this new complementary strand is synthesized in 5' to 3' direction. It is a simple process. While regarding the synthesis of this new complementary strand, direction is a problem, because the synthesis of these two new strands are catalyzed by a single dimer of DNA polymerase 3. So while this strand is synthesized in this direction, 5' to 3' direction, this strand has to be synthesized in opposite direction. Opposite direction means this strand is also synthesized in 5' to 3' direction. So one strand is synthesized in this direction and this strand is synthesized in that direction. So how the DNA polymerase 3 is coordinating the synthesis of these two new complementary strands? This new complementary strand is synthesized continuously in 5' to 3' direction in a hassle-free manner. This strand is known as leading strand, whereas this new strand is synthesized discontinuously in the form of Okazaki fragments. Okazaki fragments are the DNA fragments. These black colored patches are Okazaki fragments. These Okazaki fragments are also synthesized by the same enzyme DNA polymerase 3. And for each Okazaki fragment, there must be a synthesis of an RNA primer. This is this green color strand. This green color patch is an RNA primer. This is also an RNA primer, and this is also another RNA primer. The enzyme, while catalyzing the synthesis of leading strand, it adds nucleotide to the free 3-hydroxyl group of this individual RNA primer, thereby catalyzing the synthesis of lagging strands in the form of Okazaki fragments. So one strand is known as leading strand. Leading strand is synthesized continuously, and the other strand is lagging strand. Synthesis of lagging strand occurs discontinuously in the form of Okazaki fragments. So during the synthesis of leading strand and lagging strand, direction is a problem, because how a single dimer of DNA polymerase 3 can catalyze the synthesis of two opposing directions? We will look into that. Friends, in this diagram, let us understand various structures that are present. These are the two DNA polymerase 3. These ring-like structures are two molecules of DNA polymerase 3. So it forms a dimer of DNA polymerase 3. And this is the enzyme helicase. and this is primates. You know about their function. And these are the single-stranded binding protein. The function of the single-stranded binding protein is to stabilize the separated strand and to prevent their reannealing. So the problem that is encountered during the synthesis of lagging strand can be solved by looping of the DNA strand. Here friends, you see this is a template strand. This black color strand is the template strand. So this black color strand has been looped. So by looping of this template strand, it becomes very easy for the enzyme DNA polymerase 3 to coordinate the synthesis of leading strand and lagging strand. This red colored strand is the leading strand and this blue colored patch is known as RNA primer. Similarly, the synthesis of lagging strand is discontinuous and it occurs in the form of small fragments known as Okazaki fragments. This red colored strand is a Okazaki fragment and each Okazaki fragment must begin with an RNA primer. Similarly, this red colored patch is another Okazaki fragment which has also begun with an RNA primer. So by looping of one of the template strand, it becomes easy for the enzyme DNA polymerase 3 to coordinate the synthesis of leading strand and lagging strand. After the synthesis of Okazaki fragments, these RNA primers are removed by the 5' to 3' exonuclease activity of DNA polymerase 1. DNA polymerase 1 is another enzyme that catalyzes the removal of this RNA primer by its 5' to 3' exonuclease activity. The same enzyme also synthesizes DNA in place of RNA primer. After the synthesis of DNA, the nick which is left between DNA fragments, these nicks are sealed by the enzyme DNA ligase. So friends, this is about elongation. After elongation, next phase is termination. In this phase, the DNA replication is terminated. Friends, this is the circular double-stranded DNA molecule which is present in E. coli. The circular DNA molecule is the chromosome. And in this circular double-stranded DNA molecule, this is the ORIC. And ORIC is the specific point of origin of replication from which replication starts. So from this origin of replication, two replication fork, one move in rightward direction and the other move in leftward directions. So there are two replication forks, they move in opposite direction, one move in rightward direction and the other replication fork move in leftward direction at a rate of approximately 6 kilobase per minute. This zone, which is underlined in red, is the termination zone. The termination zone contains 10 specific sites known as tear sites. These arrowheads, which are colored in blue and black, are the tear sites or termination sites. 10 such termination sites are present in the termination zone. These tear sites are named from A to J. These tear sites along with a specific group of protein known as TUS protein. This TUS protein is known as DNA replication terminus site binding protein. This TUS protein binds with these tear sites by which DNA replication is terminated. So there are two replication forks, they move in opposite direction and at last these two replication fork meet. When the two replication forks meet, DNA replication is terminated. So this is the direction of movement of leftward replication fork, and this is the direction of movement of the rightward replication fork. The leftward replication fork can pass 5 such tail sites, but after that it cannot move further. So the leftward replication fork is stalled after passing these 5 blue sides. That means at this position the leftward replication fork will be stalled. Similarly, the rightward replication fork will move in this direction and it will pass 5 black tail sites, and after that it will not move further. So at this site, both of the replication forks which are moving in opposite direction will be stalled. So the process of DNA replication will be terminated. Friends, this is about the process of DNA replication in E. coli. Please subscribe my YouTube channel Sam Online Class, requesting you to give your feedbacks so that I can improve my performance, and also I'll be able to improve the quality of the videos so that it will be more useful for you. Thank you friends. See you. Bye bye.